Hello, everyone, to whoever's tuning in. Good morning, good afternoon, or good evening, whichever part of the world you're from. And welcome to this ION World 2022 lecture entitled Utilization of the Thermo Fisher Genexus Turnkey Next Generation Sequencing Platform for Molecular Profiling of Non-Small Cell Lung Cancer Samples. I'm Dr. Brandon Pang. I'm a molecular histopathologist from Singapore, as well as an adjunct research fellow from the Department of Pathology at the Yong Lin School of Medicine at the National University of Singapore. Without further ado, let's proceed. So these are my disclosures in terms of consultancy and honoraria that I've received from various commercial entities. And the first section of this journey will take us through the clinical background of molecular profiling in non-small cell lung cancer. That is basically acronym NSCLC for short. So whenever you see this NSCLC, that will be referring to non-small cell lung cancer. So for those of us who are involved in one way or another in translational research in non-small cell lung cancer, there are certain existing paradigms that probably most or all of us would be familiar with, and that is the necessity for multiplex testing. So there are extremely new and always expanding numbers of novel and established biomarkers that have been basically become the subject of a lot of interest for all of us. And if we refer to the NCCN guidelines, we'll see that a list of routine biomarkers would include genes such as EGFR and BRAF, as well as ALK, ROS1, and RET, which are very important for gene fusions. Of course, the NTRK gene fusion is also important, and it's a bit more novel. And what I would like to draw your attention to is the presence of this event called the MET exon 14 skipping mutations. And I will be sort of delving a little bit more into this specific mutation in the first half of my lecture, in case some of you in the audience are not familiar with its importance. We also, there is emerging interest in some biomarkers like high-level MET amplification, various kinds of HER2 alterations, as well as tumor mutational burden. On the left, the list is recommended for routine testing for lung cancer samples as and when they become available. And I will share with you why most of the world is moving into very broad molecular profiling to identify all of these actionable markers at the same time. And we are sort of slowly but surely leaving behind the idea of testing for these gene alterations one at a time, but we want to do them all together. So the MET exon 14 splice site mutations engender the process of alternative splicing. So if we look at the DNA of the MET gene as it's encoded in our genome, the occurrence of these splice site mutations, which may be close to either the 3' or the 5' end of the 14th exon of the MET gene, when that gets transcribed to pre-messenger RNA, we find that alternative splicing inevitably occurs whenever some of these mutations are present, and exon 14 is sort of omitted from the final messenger RNA copy because it's always spliced out. And when the messenger RNA becomes transcribed and translated into protein, we find that this exon 14 deficient MET sort of a receptor has a very strong downstream pathway activation of downstream targets. And the ubiquitin ligase is unable to sort of attach ubiquitin onto MET exon 14 because that is the ubiquitin binding site. And as a consequence of that, there is decreased degradation of these receptors, which then slowly but surely accumulate, and this will result to the phenomenon of MET overexpression if one uses immunohistochemistry to sort of stain for the MET exon, the MET gene product on the cell membrane of the cancer cells. So if we look at the MET exon 14 structure, you see in this region here that there is the exon with a 5' and 3' intron. And if we have these mutations occurring at the 5' intronic end, the 3' coding region, or the 3' region spanning both the intron exon junction, all of these events will lead to the alternative splicing out of MET exon 14 from the pre-messenger RNA to the final messenger RNA copy. So obviously, there are two ways that one could go about hunting for these events. By sequencing at the DNA level, one has to sort of navigate all these collections of different events that could all occur at the same time. Or, there's an easier and rapidly becoming more popular way of looking for it by sequencing the RNA and just finding MET exon 14 RNA that is lacking in exon 14, which kind of makes the job easier because one would not have to sort of be aware of existing as well as yet to be determined ways in which this alternative splicing could be triggered in the spliceosome. So MET exon 14 skipping mutations are the newest actionable oncogenic alteration in non-small cell lung cancer. But in the real world, cost effectiveness is extremely important, regardless of the kind of healthcare system that you operate in, whether it's universal healthcare or patient pays, or whether it's insurance being the payer. Cost effectiveness is exceedingly important in determining how one goes about looking for these mutations in lung cancer patients. And as I've sort of showed you in my earlier slide, it's always a lot wiser to go about it by sequencing RNA than DNA, especially when one has limited resources. We know that in cancer patients, the biopsy that the interventional radiologist performs on the patient will yield a paraffin block that is used for many, if not most, downstream applications. So this will include visualizing these alterations through microscopy by the techniques of immunohistochemistry of FISH, which can detect things like PD-L1 expression levels, as well as ALK expression, aberrant ALK expression of ALK rearranged isoforms, as well as ROS1 and MET expression. On the other hand, nucleic acid type of testing can be done in two ways, by PCR or Sanger sequencing, as well as next generation sequencing. So in PCR, one typically looks at one or the most half a dozen concurrent alterations, whereas NGS has the capacity to interrogate anything from up to the whole exome or the whole genome if necessary. And we can detect everything that might be useful right now or relevant for the management of non-small cell lung cancer, and be able to sort of perform translational research in terms of determining whether other novel and increasingly important or relevant mutations are there or not in a particular sample. So what are the ways in which one can obtain diagnostic material in non-small cell lung cancer? The first would be a fine needle aspiration biopsy, which might give approximately 200 microns worth of formalin-fixed paraffin embedded material for all required tests. Each time a block of a research archive or archive block is retrieved, a certain amount of that tissue is lost as the block is faced and trimmed in preparation for cutting a new batch of sections. So the NCCN guidelines currently recommend that many biomarkers be tested on lung tissue and include everything that I mentioned in my earlier slides, in addition to emerging markers such as tumor mutational burden and high MET amplification. So a recent study has showed that 85% of patients that are tested sequentially are still tested sequentially despite the availability of NGS as a modality for use in the diagnostic setting. And if all these tests are performed sequentially, there will be little or no tissue left for MET testing. So this is some data from the VISION study showing that in a typical lung biopsy, so imagine if this is the width of a core of lung tissue, and we start doing H&E and immunohistochemistry for the diagnostic confirmation of lung cancer, that will take up about 38 microns of material. Then we move into EGFR, which might take up 70 microns. And if we then sequentially perform ALK, ROS1, and PD-L1 testing, that will utilize another 30 microns. And we have only 60 microns left for NGS that might not include RNA sequencing or the MET exon 14 skipping alteration in the constitution of the gene panel. So if we use up 198 microns, that leaves just 2 microns for additional testing. If you look at this chart on the right, we find that the sequence in which we go about testing will influence the extent in which patients have enough residual material to sort of carry on testing for all the NCCN guideline recommended biomarkers. So you see that MET is right at the end, with only about less than 30% having enough material to complete assessment of MET exon 14. So this whole process of tissue-based mutation testing is familiar and established, but nonetheless presents challenges to us. We may only have one tissue block, or we may have multiple tissue blocks to work from. Whoever's in charge of the project has to select the best tissue block with enough material in it. If you're analyzing tumor and not tumor-associated stroma, you have to dissect it out, and that reduces the amount of starting material. You isolate and purify nucleic acids, assess their quality, and then you assess whether or not your coverage of your library prep is adequate to assess all the genes or regions of interest. And finally, you get to the last step, which is making a treatment decision, yes or no, for a particular drug. So I'll now go on with my experience over the last 1.5 years with the Genexus Oncomine Precision Assay for lung cancer. So essentially, there are two components to the Genexus system: nucleic acid purification, quantification, and plating system. So I only had the Genexus sequencer and not the purification system installed in my laboratory. So if you install both of these systems, you will only have up to 10 minutes of touch time in total. But because we did not have the purification system, we ended up with about 2 hours of hands-on wet and dry lab time spread out to about over 2 working days, because the nucleic acid extraction takes place over the first day, and then QC and qualification takes place early in the second day before the sample is loaded onto the Genexus. And you realize that very quickly overnight, by the morning of the third day, all the actionable genetic alterations in the entire batch of samples would have been flagged out by the Genexus. And I had a very good experience with this, because it was really revolutionary, and it cut down my handling time, which typically took 5 to 6 working days, and a lot of manual bioinformatic cleanup and stuff like that. So all that is done away with when one adopts the Genexus platform. So as you can see, there are about 50 genes in the OPA lung panel, which includes DNA hotspots. It has a potential to enumerate copy number variations in these 10-odd genes. It can look at the RNA level to detect intergenic fusions, as well as intragenic rearrangements in the ALK gene, BRAF, ESR1, FGFR1 to 3, and MET, as well as intragenic fusions. So this is why this platform is so appealing. There is a low input of nucleic acid required with only 10 nanograms of either DNA or RNA, and that leads to a high success rate for the utilization of this assay. From my mind, from my experience, less than 5% of my research samples actually turn up with an unsatisfactory result, and that certainly agrees with everything that Thermo Fisher have put up on their website pertaining to the Genexus system. And the system is slated to be CIVD certified sometime this year, so do stay tuned for updates. This is a small series of 15 samples that I run on a little study that compared 3 platforms. So I used to use the Archer system, both FusionPlex and VariantPlex, and I had some archival research samples which I then ran on the Genexus. As you can see, the top 10 samples were entirely concordant for ALK, MET, RET, and ROS fusions, as well as MET exon 14 gene rearrangement. So there were 2 cases that I ran, and they were both concordant. And I then ran 5 cases, archival samples, which included 3 MET exon 14 skipping events on the Genexus, as well as this other platform called the Idilla fusion cartridge, and they were all concordant as well. So that showed great reproducibility between 3 platforms for some of these most important oncogenic alterations that are known to occur in non-small cell lung cancer. And there's a much larger series that Idilla have published on their fusion cartridge website. So please look up this study if you're interested as well. So the Genexus can run both tissue as well as liquid biopsy. So a theoretical dual liquid and tissues sample workflow could look something like this. On the first day when the sample is obtained, so the touch prep can be examined. If it's adequate, that can immediately go for nucleic acid extraction. If that is not good enough, then the tissue alone can perhaps be used for PDL1, and the liquid biopsy can be drawn from the patient, and that can be put through the Genexus. And we can get back all these results, no matter what you do, by the 3rd or the 5th day, where we give 2 days allowance for another 2 or 3 more samples to be run as a batch together with that first sample. So otherwise, if there were enough samples, if there were 3 to 4 samples a day, you could actually cut this down to 3 days. So I mean, I hope that those of you who are thinking of adopting the Genexus system will basically share my positive experience with Genexus, and good luck for all your research projects. My last slide. Thank you very much, and enjoy the rest of the event, Iron World 2022. Thank you.